Yeah, he was.
Okay, everyone, we're about to get started. Okay, very, very good. Welcome to the Harvey Government Center here in Key West, Florida. It's March 15, 2012, for those who are looking at this on video or DVD. My name is Michael Doyle, and I'm the Executive Director of the Florida Keys Mosquito Control District. We have several other people here with us I'd like to introduce. On your right is Hayden Perry. He's the CEO of Oxitec.
Thank you.
Luke Alfie is the Chief Scientist and Founder of Oxitec. I'll be in the middle there in just a moment. Then we have Colleen Fitzsimmons. She is a biologist and the public relations person for the Florida Keys Mosquito Control District. And Dr. Mark Whiteside, he's the head physician at the Monroe County Health Department, and he's the person that we work with directly for all what we need to know about dengue in this area. All right. I need you to know that this entire presentation and the questions and answers afterwards is being recorded, both by some documentary folks, both from England and the United States, and then it is also on the local cable news station, and it will be recorded onto a DVD. So if you're interested in getting that at a later point, go to the Florida Keys Mosquito Control District website, click on Contact Us, and there'll be an email there. I think it's help@Florida Keys Mosquito Control, or at keysmosquito.org, and then you can get a copy of that DVD. Just request one there, and we'll make sure you get one. All right, it's a good-sized crowd we have here, and I'm just in terms of my presentation, I'll be doing some presenting for folks that are outside the Keys. I'm curious how many people here are from the Keys by show of hands, if you don't mind. Very good. And then how about from Key West proper, the city of Key West? Also, all right, large number, very good, thank you. The purpose of the meeting is really threefold. One is to present information to the public to help form an informed opinion about this issue. The second is better for us to understand the questions and concerns that the public has about a new technology like this. And the third is to answer as many questions as we can in this short time. We have just until about 7 o'clock for this, so we're going to have about a half-hour presentation, half-hour to 40 minutes, and then we'll open up for questions. Speaking of questions, you may have gotten a survey from North Carolina State as you came in, and if you're finished with those, if you could pass those to the outside. Do I see anyone here to pick them up? You can. Pick them, pass them to the outside, they'll be picked up, and then I understand if you're interested in doing a exit survey, that will be available also. Okay, anyone from staff, anything else I need to mention? All right. You know, before we get started, let me mention our staff here. If you're wondering who all the folks are here. To my right is Andrea Leo, she's the deputy director of the district. Next to her is Mickey Koss, who supervises all the domestic inspectors here in Key West, so she's in charge of basically all the mosquito control in Key West. Next is Catherine Brzezinski. She's a PCR biologist who works for us primarily in Marathon, but does a lot of the surveillance work also down here in Key West. In fact, the majority in the lower Keys also. And next to her is Dr. Larry Hrebar. He is our senior entomologist out of the Marathon office. And then we have Juliana Waldrop, and she's the executive assistant there. And you'll be seeing John David. John, I lost your last name for a second. Snell, thank you. John David Snell, he'll be assisting with the question and answer and when we get to that point. All right. Let's begin with my presentation then, and then we'll move on to Oxitec's. Okay, first, to get everything started, I wanted to do a quick one slide. Here's the status, and then we'll go into the background. So we'll go forward first, then back up with the background. The current status of the project is we have no genetically modified mosquito eggs or adults in Key West at this time, and have not in the past. The release date has not been set, and that depends very much on the decisions by the state and federal agencies that we're working with. The release area, the size and location may be determined by the agencies, so we have 2 or 3 areas in town that we're strongly considering, but those have not been defined exactly yet. The number of mosquitoes to be released, that's a big question we've been having, and that is, I'll explain, I have a good slide towards the end of the presentation at the Q&A section, and I can give you more details, but just for the preview is, that really is known several weeks before the trial would begin, because it's based on the local mosquito population. And finally, I'm happy to report that we've had no dengue reported in Key West since November of 2010. Okay, quick background for those who are not from the Key West area. So I'll move through this quickly since most of us are from Key West. Key West is the most southwestern island in the Florida Keys, about 1,000 islands in the Keys, and we're about 120 miles south of Miami. We have about 4 by 1.5 square miles in the city. The majority of land, let me back this up for this slide. This is the Keys for those who aren't familiar with it. The stars on that represent where the mosquito control district's offices are, one in Key West, one in Marathon, and one in Key Largo. It's about 100 or more miles between those, so it's one of probably the longest district in the country. Okay, back to Key West. The majority of the island of Key West is densely populated, and you can see here from this aerial photograph that the majority is urbanized area, and there was one section over here that's the airport and surrounding lands there. So it's a pretty ideal location for the Aedes aegypti mosquito, which we'll talk about in just a minute. Key West background, latitude is 23.5 to 25.5. We're definitely tropical, 80 to 90 degrees in the summer, 70 to 80 in the winter. Rainy season is basically May to October, which has an impact on the mosquito populations, and in my opinion, it's nice all year round. The population, permanent population, is just under 25,000 people, but the estimated visitors per year is about 3.4 million. So the point just being that a lot of people come in and out, and that has a big effect on any mosquito-borne issues. Mosquitoes that we have, yellow fever in the past was a big issue here in the 19th and early 20th century. In fact, Dr. Porter from here, who is known to be like the yellow fever expert of the United States, is from Key West. The last known epidemic was 1936, and that was dengue here until 2009-10, where we had that resurgence. And again, no reported cases since November of 2010. A little bit about the district. We were established in 1951, and our mission is to protect the residents of Florida Keys from mosquitoes and the diseases that they carry. That is why all of this is happening. We're headquartered in Key West, as you saw, with aerial facilities in Marathon and in Key Largo, our field office. We have a large staff for large numbers of mosquitoes. There's 78 full-time staff, including several biologists, many of whom you've met right here. And then since the dengue epidemic had started in 2009, we ended up hiring 10 full-time staff just for this issue. So it's a big part of the program now. A little bit about the mosquitoes. There's 48 species. Thanks to Dr. Freebar, we'd found, 3 more since 2002. But the vast majority are in mangrove swamps outside of Key West. There's salt marsh mosquitoes, Aedes tenurensis. There's 2 primary species in Key West, and those are Aedes aegypti, which you'll hear quite a bit about tonight, the yellow fever mosquito, and QX quinquefasciatus, which is a southern house mosquito. And I should point out that you'll notice that those 2 primary species are both very urban species, as you can see from the name of the second one. As a visual of that, the black salt marsh mosquito, tenurensis, makes up 94% of all the mosquitoes in the Keys. All the other species outside of aegypti make up about 5%, and this Aedes aegypti, oh, that's red, isn't it? That's 0.8 of the total mosquitoes, that little tiny red sliver, if you can see that on the screen. And the reason I brought that up is that less than 1% of the mosquitoes that we have here cost more than 10% of our annual budget, so it's costing over $1 million of our budget. And that's ongoing pesticide costs that we're buying pesticides weekly, year after year for those treatments for aegypti. We have 10 plus full-time personnel who are dedicated primarily to aegypti, either monitoring or control, and then the cost of the aircraft that you see every few weeks. So all told, it's a very expensive proposition to try to keep Key West as safe as it is. A little bit about Aedes aegypti, in fact, a lot of bit about Aedes aegypti, is that it's an urban mosquito found exclusively near humans. It flies only a few blocks. Only females bite. Most of us are aware of that. They live for a few days to a few weeks, so they die quite quickly. About 10% or so die every day with the wild ones. Adults hide in and under homes, which is why they're hard to treat. The larvae are found almost entirely in containers and debris around humans. They're a human-based mosquito. Anywhere humans are in tropical areas, you often find them, and where humans aren't, you will not. Larvae prefer clean water. Of the research that we've done, 99.9% of larvae are found in non-sewage related water. I believe Dr. Rebar told me it was in 2002 when we did an extensive survey, it was 2 locations that had sewage type water out of 4,777. Likely a very small portion of an urban predator's diet, and mainly because there aren't that many of them. They're not much biomass there compared to beetles and moths and other larger prey species. They are invasive to Key West. They arrive by ships, planes, trucks from the mainland and the Caribbean several hundred years ago and been here ever since. Very efficient vectors of yellow fever and dengue, and that's for 2 reasons. I'm an entomologist, so this part's pretty exciting to me. They're physically and behaviorally a good vector. They prefer to bite humans. That's their main prey, or host, I should say. The adults are what people call snack biters, not a very official term, but they bite they might bite multiple people at a time. They prefer that versus one large blood meal and then back into the bushes. And they bite primarily during the day, which is not the time that most mosquito control is done. And at present, here in the Keys, the only available control methods are dumping containers, spraying adults, and treating the larvae with larvicides. A little more about their behavior. You can see here that they will peak in June to October. So you don't see many during the winter months, and in the summer, when the rains come in May or so, their numbers skyrocket. And so that affects both control and any type of proposed release like this. What have we done? Ground larval control. Mentioned we have 9 full-time inspectors doing control, 8,000 properties, 260 houses inspected daily, 550 containers treated daily. Most houses are inspected monthly. And given how fast that they go from egg to adult, ideally, we'd want to inspect them every 7 to 10 days. There's not the funding and the people to do that. So we use long-term products wherever we can. But we would really need 40 to 50 inspectors to hit every house often enough. And so you can see sort of the challenge of doing control for a species like this. So given the fact that it's hard to get to all those places, we started doing helicopter control in February of 2011, just about a year ago. All of Key West is sprayed about every 2 weeks during the raising season based on our trap counts. Everything we do is based on trap counts. It costs about $25,000 for every one of those spray treatments. So you can add it adds up very quickly. That is very good at killing mosquitoes in thousands of these above-ground containers. But something unique about Key West, in many older cities like this, is the cisterns underneath homes. They are good producers of Aedes aegypti, and it's very difficult for the inspectors to find them because they're often hidden, just a crack or a tube sticking out of the ground. They're hard to find them. And then the helicopter, as good as it is for all the small treatments, is very difficult, impossible for that type of treatment to get to the underground containers. We in fact, just last few days, been working with the helicopters to improve that treatment, get better droplet size, and that type of thing. So we're hoping that we'll do much better this coming year. But still, it's a tough and expensive process. So we also do a ground adult spraying. Almost 1,300 yards were sprayed last year with a general spectrum pesticide that does kill most small insects. But it's very good for Aegypti because it gets into the hiding places. They love to hide underneath things and in houses. And so that's the treatment, and it's very time consuming. Also done quite a bit of public education. I won't go through all this, it's a little overwhelming with all of it, but trust me, it's a very intensive effort to help encourage the public to clean yards, take personal precautions. That's been going on for almost 2 years, and that whole list, you can look on that DVD later, but Colleen's been very busy the last several years with all of this, trying to encourage the public. Not only us, but Monroe County started the ABCD program. I'm sure, according to a recent survey, about 80% of the people are very familiar with this. There were door hangers and newspaper articles and cleanup weekends and newsletters, speakers, water bills, comic strips, Stan Dingwater, one of my favorite characters, and radio spots to teach us all about how to break the cycle of dengue. And very many people know about it. 82% said they had done something since May. This survey was done this past summer to get rid of water on their property. Taking action was associated with the events, the outreach we did. But unfortunately, with a difficult species like that, out of the 55 properties that were surveyed in that particular study, 91% of the residents said that they were taking care of water. So people were attempting to take care of it, but 9 times out of 10, there were still Aegypti being produced on those properties. So it takes a lot of work to find all those places. Again, that's why it's been a tough mosquito to control anywhere in the world. So what are the results been? This is a combined efforts in 2011. The simple part of this, the number on the bars represent the number of containers, percentage of containers found in Key West on that particular day by our domestic inspectors, and this is the peak of the season from August to October of 2011. Our goal is less than 5% of containers producing mosquitoes, and you can see that it's probably double that if you average all of that, not including the weekends. So given the fact that, in my opinion, this is one of the most advanced programs in the United States, and anyone who does mosquito control would say we're definitely one of the best, it still doesn't appear to be enough. So what we're looking for is a tool that is more effective in terms of reducing the total population, less expensive, and less pesticide dependent, quite honestly. All right, that's my presentation. So now Hayden Perry of Oxitec will step up to do his.
Great. Well, thank you very much, and thank all of you for this opportunity to talk to you. We're a young company. We're pioneering a new approach, and I think that needs explanation, and I'm delighted to have this opportunity to be here today. So a little bit of background about us and then what we're trying to do. We are a UK company. We're founded out of Oxford University, and in fact Luke over there is the founder and the scientific brains behind the company. And he's been working on this probably for 5 or more years before a company was formed. And then we've been developing this approach for the last 10 years. We're totally private, so we're funded by private individuals, private shareholders from around the world. We have US shareholders, Brazilian ones, Asian ones, and some European ones. Just to counter one story, we have no corporate investors, so there's no big multinational who invest in us in any way. So it's all private individuals, and we're a small company, 40 staff, and we're working, as you can see, in quite a large number of countries around the world now. And how these collaborations start really is that we find that often it's Luke will present at a conference about what we're trying to do, and then either a Minister of Health or Head of Agriculture will come up and say, can we have a go at this in our area because we've got a particular issue. And so we start a collaboration. We always work with a leading institute in any country. We're never on our own. And as you can see there, we've got US Department of Agriculture, Ministry of Health, Malaysia, the Gorgas Institute in Panama. These are the kinds of people we're working with. We've had quite a lot of international recognition as well, from people like the Wellcome Trust and the World Economic Forum, and also Grand Challenges in Global Health, which is a program funded predominantly by the Gates Foundation. And I mention that not because I wish to boost ourselves up, but it's just all of these organizations on there do carry out very intense due diligence on us. They're very serious scientists. They're passionate about what they do as well as we are passionate about what we do. And so we're put through our paces by a lot of people in terms of the quality of our science. So in terms of what we're trying to do, we're basically trying to control insects. Now, insects are a major threat to the food crops that we grow, and insects are a major threat to human health. Insects spread disease like malaria, dengue fever, which is the subject of some of today's conversations. And we've got a way, a philosophy, a concept, an approach by which we want to reduce the insect population that's causing the problem, whether it's eating your crop or whether it's spreading disease. We want to do so in a way that's safe to humans and environmentally friendly, very low impact. And that's actually very important. So dengue fever, let's a little bit about dengue fever. We've now got about 50 to 100 million cases around the world, according to the WHO. Spreading very fast. It's increased about 30-fold in incidence in the last 50 years. Spread predominantly by one insect that came out of North Africa and then has spread itself around the world. And what happens is the insect will come in, it'll be resident in a town, a country, and then when someone comes in carrying dengue fever in their blood, then the insect will bite them, then they'll bite somebody else, and so dengue is spread. Now, dengue fever, the symptoms of dengue fever can vary tremendously. You can get asymptomatic, where you don't feel any symptoms at all, through nausea, headache, sickness, and then you get the more severe forms of hemorrhagic fever and shock syndrome. Now, a particular peculiarity of dengue is that there are, in fact, 4 different types. And what happens is, if you're bitten by a mosquito carrying dengue and you develop symptoms, then your body will produce antibodies, and when you're bitten by, on another occasion by a mosquito carrying the same virus, you'll be resistant. The difficulty is that when bitten by one of the other 3 strains, then you are more susceptible, and then the symptoms will tend to be more severe. So it's a peculiar one because not only is the incidence growing around the world quite rapidly, but the symptoms get worse. Now, there's no medication at the moment, and there's no vaccine at the moment. And there are companies trying to develop vaccines. The most advanced is from Sanofi, and that's actually has something in common with us because actually it's a genetically modified vaccine. It's using the sort of same basic concept as we use. But they're not in the market yet, they're not being used yet. And it's the same mosquito that's spreading another disease called chikungunya and then yellow fever. It's the same mosquito. And the bars, you can see the 2 lines, you can see basically the zone around the world where Aedes aegypti inhabits and then where you get dengue fever. So it's very widespread. Now, as Michael said, Florida Keys had no dengue fever in 2011, which is excellent. but what you see around the patch is basically, over time, you get more and more cases of dengue fever reported. So what I've shown here is basically the area to the south. It's the area of the Caribbean and Central America, where you've got a rising trend of dengue fever, and you can see in the last sort of 5 years or so it's pretty much doubled. Now, probably not so visible, but I've tried to put it on there, is the global trend, and you can see it's following the same pattern. Dengue is increasing in terms of the total number of cases. And the reason it's growing is because of this mosquito. At the moment, the only way to control the spread of dengue is to control the mosquito. So dengue prevention is solely a question of mosquito control currently. And this insect is very difficult to control. It's an invasive species in most countries, including in Florida. Shouldn't be here, brought in by man. It's very well adapted to the urban environment. Breeds in little containers, clear water containers in and around the home. So you basically got to get all the containers. And it lives in and around the home, doesn't travel very far. So it means that the control concepts that one needs to employ is you've got to have a lot of boots on the ground. It's a very intense control program. And even the best teams in the world struggle to control it. So our solution is this, and I've tried to express this in a graphic form. Basically, with mosquitoes and Aedes aegypti here, male will meet female. The female will lay up to about 500 eggs in her lifetime. The female is the one that bites you and spread disease. The male can't bite. It doesn't have the mouth parts to bite, doesn't want to bite you. So it's the female that's representing the main danger here. So if we could take this female out, if we could stop this female actually producing so many eggs, then we can have an effect on reducing the population of that insect. And that's the philosophy here. So what we do is we release male mosquitoes, which don't bite. Now they contain a gene which actually prevent the growth and the survival of the offspring, the next generation. So we'll send out the males, like most males, biologically programmed to look for females. They'll mate, the female will lay eggs, and they won't survive up to adults. If we can put more males out there that are actually in the environment, then the females will be more likely to mate with ones that can't produce viable offspring. It becomes a numbers game, and then we can bring the population down. And that's the concept. Now, here's the difficulty, which is we need to produce quite a lot of these mosquitoes, basically to outnumber the males that are already there. And that would be quite difficult to do if we were using standard techniques. So what we do is, in addition to the gene that actually prevents the offspring becoming viable adults, is we put a way to deactivate that. So in a production environment or in a laboratory, if we feed the insects with a chemical called tetracycline, it actually acts as a blocker. So we can produce very large numbers of these insects from generation to generation. Then when we have the generation that's actually going to go out into the wild, they don't have any of this tetracycline. The inability to reproduce then becomes reactivated, and that's the basis of the system. So we can produce large numbers with the tetracycline, and when you go out into the open environment, they have no access to that. And I'll come back to this point in a minute. And so they will be infertile. They won't produce. They will produce offspring. They'll produce larvae, but they won't emerge into viable adults, and we can get that population down. I should say that this is a system that I think is very well adapted, can be blended in with normal control procedures. So a lot of mosquito control focuses on larvicides to control Aegypti, and this is very compatible with that. So I think in practice what we're going to see is we're going to see control departments using this approach in conjunction with other measures to actually control the problem. Now, in many ways, this isn't new. So in many ways, this isn't new. Actually, what this is a new twist on an old story. because we've actually been using, and I use the sort of wider term of we, mankind, we've actually been using sterile insects for about 50 years now. And this has been pioneered here in the US, and it's actually what was the driver, the base concept behind what we're doing. So what's been happening for the last 50 years is particularly the USDA have been using radiation to actually radiate insects to the point at which they are in effect sterile. They won't produce the next generation, but they're still fit enough to go out and mate. And that was started with, there's been a number of different programs. I think the first release of one called the New World screwworm, which is a nasty insect that lays its eggs in the wounds of cattle. That was actually in Florida. We've also got there is also a Mediterranean fruit fly program here in Florida, and that's actually supplied from that factory in Guatemala. So in practice, it would be a really good idea, and people have tried for many, many years to try and get this to work in mosquitoes, because mosquitoes are a very natural target for this approach. You just want to take out the one insect that's causing the problem. But it's not really proved viable, and that's because it's actually very difficult to give the mosquito the right amount of radiation dose to render it unable to produce offspring without actually damaging it so much that it's not fit and healthy. And so you can sterilize the mosquito, but when they go out there in the wild, they're not actually competing with the wild males. And that means that whereas this sort of philosophy, this approach, has been used in agriculture in quite a few occasions, it's never been proved possible in mosquitoes before. So this is basically the foundation for Oxitec's technology, which is if we can do the same thing, but using a single piece of DNA to have the same effect in a very targeted manner, then we can actually produce the same effect, very targeted, very great precision, and we don't have the fitness penalty in the insect that makes it incapable of going out and mating. So we've done this in mosquitoes, we've done this in a number of different agricultural species. And what you see is it's very effective, they mate very well, you don't have this fitness penalty, and we can do this male-only release. So we can release the insect that's safe, if you like, and not release both male and female together, which radiation tends to do. So the benefit in what we're doing, in the way I see it, is we're producing a new way, or a new twist on an old way, of being able to reduce that target insect population. It's species specific. Now mosquitoes, Aedes aegypti, they only mate with Aedes aegypti. They don't mate with other species. So whatever you do in the Aedes aegypti, it's not going to cross over into other species. It's going to stay where it is. We call it a self-limiting strategy, which is a bit of a mouthful, but effectively what we're putting in there is an inability to reproduce effectively. So it is limiting. You're actually putting in a way of stopping something happening. You're not putting something in that's going to replicate through many generations and that's going to spread. It's stopping it. So it's very controllable. So let's assume, for example, that we're releasing some mosquitoes in an area and bringing that population down, and for whatever reason one wanted to stop. Then those mosquitoes are just going to die. I mean, they die after a few days. That gene gets completely eliminated from the environment, and you're just going to get the wild mosquito population coming back up again. So you're not putting in a train of events where you're spreading a gene through a population. Now, the other issue here, in terms of control, is these mosquitoes don't actually fly very far. Each mosquito will fly maybe 100 yards, up to 200 yards. They don't spread around. So actually, if you release a whole series of mosquitoes in one place, then they're going to be within a certain radius. So you know where they go, you know where they are. So you're not going to have a knock-on effect. So we shouldn't think about this as a method for controlling Aedes aegypti in sort of huge territories and wiping out dengue fever. No, we should think about this as, I want to control this insect in this area. Now this area could be a town, it could be a city, it could be an island. But you always need to think about a defined area. And each mosquito, what's it going to do? Each male that we send out, it's going to go out there, it's going to find a female mate and there's no offspring, or it's not going to find a female. In both cases, it's going to die, the offspring are going to die. It's a finite end. And in agriculture, not mosquitoes, but I've put up a comment there from APHIS, which is the USDA, is that when this was looked at in agriculture, thinking about using fruit flies and a cotton pest, after deliberation of the impact on the environment, looking at this method as well as other methods of controlling insects, this method was seen to be environmentally preferable. In other words, a lower environmental impact than other solutions. So the releases, just sort of give a mental image of how this is going to happen. What we would do is we would actually produce the male mosquitoes, take them into the environment, and release the males. And so you release them in an area, and they'll fly up to 200 yards, seeking out the females. And the sort of release you can do is, this is one of our guys. This is actually a picture from him releasing in Brazil, where we're working at the moment. So he takes the cage out, takes the top off, the males go out. And then he goes a bit further down the street, because you've got to have overlapping control, and releases them again, and those males will travel that distance, and you go further down the street again, and it's the same idea, until you've covered whatever zone you want to cover. Now, I'd just like to go back to this tetracycline question, because there's been quite a lot of discussion, I think, in the media that says, okay, well, these guys have got this system for tetracycline that will restore the fertility, which is what they do in the lab, but what happens if you find tetracycline in the environment? And so there's been quite a lot of debate about the levels of tetracycline in the environment, whether it, how it breaks down, etc. And we're very confident that you won't find tetracycline in the environment in the right conditions, at the right time, in the way these mosquitoes need. But that's actually not, I think, the most important point. The most important point, I think, is actually, let's assume, for the sake of argument, purely hypothetically, let's assume I actually go out into the street with water, with tetracycline in exactly the right amount or exactly the right conditions, and put it there so that the mosquitoes can actually have access to it. So there's a point source. Well, in that case, those mosquitoes that actually lay eggs around that, and where the larvae are grown up within that tetracycline environment, yes, they'll have their fertility restored, but they'll be flying within a zone, and they'll be quite easy to control through whichever method. So it doesn't actually invalidate the approach at all, because the mosquito doesn't actually fly that far. I'm sure we'll come back to that question in a minute, but I did want to make that point. So, where have we done this before and what happened? This is a graph that will be published, or a version of it will be published soon. It's not been published, but I did want to, because we were coming here today, I wanted to put it up here. So this is actually the what happened in the Cayman example. So it's quite a complex graph, but basically you've got time along the bottom. So we actually started releases just before July and June. Now you've got two big lines, the blue line and the red line. So the red line is basically the area in which we were making releases, and the blue line was in the neighbouring area in a village, which was the comparison area where we weren't making releases. And basically, once the rains come, the mosquitoes come. And so on the left-hand side of the graph, as the rains come, the mosquito numbers rise. And then we started releasing our mosquitoes, first of all with just trial amounts, just practicing, and then in around July time, we started releasing the right amount. About a month later, you start seeing the blue line and the red line separate, because obviously the mosquitoes are mating with females, and by October, the population was right down. So we got an 80% control between really starting in July through to October, which is a very good result. And I think that's very similar to what we would expect to see here. we'd have a trial period, and then you would go for what's called suppression. We'd have a release area, comparison area, and that's what we'd expect to see. So public engagement, we take this very, very seriously. We're working in a number of countries. We're always guided by our partners, whoever we're working with. Different communication forms work well in different countries. So we're guided by what we do, but we do take it very seriously. And as I said, I'm delighted to be here today. I think this technology is new. It's much easier to explain it face-to-face in many ways. We're not a big company with huge budgets. We can't do big advertising campaigns. And I think the more we can get in front of people and explain it, the better it is. So thank you very much for listening.
Okay, thank you, Hayden. Now it's for the next 45 minutes or so, we'll be able to ask questions. Here's the general guidelines. Since we have so many folks that are interested tonight, you have signed up on this sign-up sheet. I have those here in front of me. I'll just call people in the order they were signed up. First come, first serve. Looks like we have about 14 people who wish to say something. Each person will have 3 minutes. John David will has the microphone there. So what I will do is I'll say your name, and then just put your hand up, and he'll come right to you. In fact, I'll mention the person after that too, just so you know who'll be coming. We'll summarize them, and then this is in terms of the questions. Everyone, the questions that are asked that I have here, we'll summarize those questions and then put those on our website for a longer answer, because we may not get to everyone's, but I want to make sure everyone's questions are answered. So we'll put those on our website in the coming weeks. And then also, any questions we get will be submitted to the state and federal regulators also, to make sure that they include those things in their evaluations also. All right, let's see. Just to mention, I know I don't want to take any more time, but I have a bunch of other information about timing and things like that, so we can bring some of those things up as we go along, if it helps to answer those questions. All right, first I have the first person is Midge Jolly, and then after her will be Jules. Okay, there's Midge right there. Go ahead. At the end of the 3 minutes, we'll move on to the next person. Thank you.
Thank you for coming. My first question is, there are members of the community who could not be here tonight, and they want to know, is there going to be a forum for them to present questions that would then be submitted to the state regulators?
I'll handle that.
I was just going to say, they're welcome to email us with these questions, and we're going to forward them on to the regulators. And not only that, like Director Doyle mentioned, we're also going to put them on the website so that they can get an extended answer to their questions. Absolutely.
Beautiful. Thank you. I have several questions, and I want to make the best use of my 3 minutes.
Okay, go ahead.
I would like to ask the gentleman from Oxitec, please, do we have numbers yet on dengue post-suppression? Not number of AGIS, but dengue post-suppression. Do we have those, and will those be made available to us?
No, shall I say that?
Go ahead.
Okay. I guess you mean in the Cayman Islands?
Wherever.
Okay. So, the suppression data that Hayden showed was from the Cayman Islands, and there, the aim of the Cayman Islands Mosquito Research and Control Unit, which is the government agency there, the equivalent of the district here, is to suppress Aedes aegypti as a preventative measure to reduce or eliminate the risk of dengue transmission. There is actually very little dengue transmission in the Cayman Islands.
Thank you. That answers my question. Please understand, I don't wish any disrespect. I trust in y'all's best intent. I trust you'll understand mine. Let's see. Can this trial actually be halted by public input, or will the only thing that will halt it be regulatory refusal?
If you want to, Gil, why don't you tell your experience in other places, and then I can jump in if needed.
Okay,
Oh,
well,
no, I really want to know about this country,
In the
because
United
if it
States?
hasn't been done here, that's the answer to my question, with all due respect.
Certainly, certainly. And the answer to that question is you mean if, just for public opinion, in terms of how that would be gauged? There's-
Is the only thing that will halt this trial regulatory refusal, or can public display halt this trial? Can our voices be heard enough
There's
that
cer-
this matters?
The voices will certainly be heard in that process, both by myself as a director and from the regulatory agencies, that if there's information that's brought up that's sufficient to show that there's a risk to this, then yes, it'd be halted either by regulatory or by myself. Yes.
I see. My next question is, does it not seem that no cases in Key West of dengue since 2010
Yes.
would suggest that our efforts, albeit emptying thousands of containers,
Right.
are successful to some degree? And how can it be better to introduce a man-made genetic aberration rather than emptying pots of water? I mean, I saw all the things about the cisterns, but it appears to be successful at this point.
It does appear that we don't have any reported cases. Is that the 3-minute point?
Yeah.
Okay, so I'll answer that. Okay, I'll finish that and go to the next person. The goal is to prevent disease issues now and in the future. So we're looking for tools. Yes, we haven't had dengue for over a year, and that's tremendous. But we had dengue version 1, that was your dengue serotype 1. If serotype 2, 3, or 4 come at some later point, in my opinion, we need a tool to immediately suppress the mosquitoes. So it's working, but in my opinion, is it good enough for our purpose and our mission here? No. Okay. Now we have Jules. Can you put your hand up, please?
Hey.
Jules.
Jules, Jewels.
And then Megan Morrison will be after that. Thank you.
All right. I'm going to kind of address the medical issues because I suffered from a very bad disease as a child. My disease is an autoimmune disease, and this was back in 1972 when I was diagnosed at just about the age of 5. I've spent a lot of my life in this wheelchair and everything, and my greatest concerns are that I moved into this area to better my health. I did not move into this area to be an experiment. I am first generation to my disease, dermatomyositis. I would like to address the issue with the physician that you have on the panel. Why are we being the guinea pigs in Key West when, as your document showed, your cases of fever are primarily in Africa, Central America, and other places around the world? I'm just wanting to know why we were chosen as the place to do this study, because, I mean, think about it. You've got a lot of people that come down here to die. You have a lot of people that are either terminal or have very severe diseases. And that's a very sensitive issue when you're talking about treating with a genetically modified organism. Once you modify those mosquitoes, you're unable to go back. You are going to alter every chain of the food line. We will be affected.
Do you want to address the medical issue, and then I've got a slide about why Key West.
Well, dengue itself, of course, can be a serious disease, and Key West is one of the first places that dengue returned to this country. We've had some cases transmitted in South Texas, for example, but we had not had cases of dengue transmitted in Florida, for example, for decades. I would emphasize this is a very limited outbreak. We had 27 confirmed cases when it was recognized in 2009, 66 confirmed cases in 2010, and as you've all mentioned, none in 2011. We've had, wonderful control programs, but we certainly don't want dengue back. Another point I would make, it was pretty much limited to Key West, Old Town, some limited spread into Newtown where I live, but not up the Keys. Aedes aegypti up the Keys, but no dengue. Most of the dengue has been, down here. Everyone recovered from it, but dengue has the potential for worse disease over time, and people with underlying conditions could certainly be at risk from especially severe forms of dengue. I guess that's the way I would answer it. Not to get into the issues with the genetically modified mosquitoes.
Okay.
But that's very important, though, in and of itself. Genetically modified organisms alter, you know, the way in which we live. So it really needs to be addressed. You've got so many transients down here, and you have a lot of tourists, and they're going to spread it all over the world because they come from all over the world to come here. It breaks my heart.
Thank you for your comments. I just put up one. I kind of anticipated that question because I've gotten from quite a few people. Thank you. So I had just a few reasons up there. But let's move on to Megan Morrison, right here, and then after that would be Heidi Golightly, up here in the front.
Hello, thank you all for having this open forum this evening in an effort to try and, for all of us as a learning experience, to try and get some intelligent conversation and dialogue going on this issue. I'm here not only representing myself, but I'm a board member of the Florida Keys Environmental Coalition. This organization is a representation of non-governmental organizations protecting the environment of the Florida Keys. Therefore, I'm a little nervous because I'm not expressing necessarily all of my own views, but I'm speaking on behalf of other people and agencies. There are a few main points that were brought to light or brought to my attention and things that I've thought about as well. Currently, there's no regulatory agency even created on a local, state level, or federal level to address this modified species. Now, genetically modified, by its terminology, indicates that it's changed. So the species is therefore changed, which indicates to me that it's not the same species, which indicates to me also that this could be an invasive species once released. And we have enough of those here already. Secondly, I haven't seen enough third-party objective research to really substantiate the claims of success that you've had in other countries. This is a proprietary, patented product. Who else has had access to research your product without doing wild experimentation? I understand that there's been research by collaborators, but collaborators by definition is not an objective term. I'm really talking about independent third-party objective research. I will stop there on that one. So that was point number 2. Point number 3, myself and others feel that this is somewhat of an ultimatum situation here, in the sense that we're faced with, why do we feel as though we're being presented with employing toxic chemical pesticide sprays or employing a genetically modified mosquito? What's behind door number 3? Can we slow down a little bit? You know, until we get the regulatory controls in place, until we learn more about this company with a proprietary patented product with a profit motive involved. We'd just like some more answers before the Mosquito Control Board makes a decision on our community's best interest and behalf on a public safety, public health and safety level, and on an environmental level. Not just the bees and the birds and the butterflies and everything, the bats, the carnivorous plants, anything that feeds on mosquitoes in the food web, in the wild, but on a public safety level as well. We have so many unanswered questions that I don't feel that all of my questions are being answered, certainly. And the community members with whom I speak are also not having their questions answered yet.
Thank you. Well, we'll get to more of those right now. Thank you. Next is Heidi Golightly. Oh, right next to, okay, very good. And after that, Joel Biddle.
First, I'd like to say I've never been to a community meeting where we weren't allowed to go up to the podium and speak. This is very intimidating and very unusual, and I've spoken a lot at different community meetings. Second, I'd like to say that above the door when I walked in, I saw Wilhelmina Harvey, and it breaks my heart to see Will, and I'll get choked up because I loved her to death, and I could hear her saying right now, this breaks my heart to think that you guys have the nerve to come here and do this to our community and to the people of Key West. It hurts. I just authored a book called Healthy Choices for a Healthy Life, and here I am sitting here listening to people tell me the place that I love and I live is going to be altered by something that I put in my book to stay away from. Anything genetically modified should not be touched in what form whatsoever, any form. And here you're telling me that I have no control because I have a feeling that this is run just like different city meetings I've been to. The minds are made up. The decision's made. These people are going to do it. You're going to approve it, and you're going to approve it. I know it. I know. I can just sense it. I can feel the vibe. This is all for naught. We're all sitting here coming with our best interest and our community's best interest, and it's just to hear this man tell me, I think we are going to see. I think. I heard that from your mouth several times. I think. If you think and don't know, because I don't think anybody knows when you're dealing with science and nature, nature's going to win out. And you know, what you've said, you could tell me all the fear stories you want, but we have somebody in this room that suffered from dengue fever, and I hope he gets to talk because he's a witness to what it is and how he felt during it. So I just think you need to think long and hard about what you're doing, and hopefully you didn't make your decision already, because it'll be a fatal mistake to the rest of us.
Thank you. Thank you very much. Staff? Okay. Joel Biddle.
Starting now. Oxitec has been criticized in the Cayman Islands, Malaysia, and Brazil for secrecy and non-disclosure. There are reports that Oxitec led citizens to believe that the release would occur in unpopulated areas, but releases were apparently made in populated areas without the residents' knowledge. You know, it's the secrecy. This is here. You've been planning this for over a year, and you're planning it. You're not thinking about it. You're not researching it. You're planning it. I find that this secrecy is, I mean, as a taxpayer, you know, you come on my property, you know, you put OV cups down, and yet it's part of a survey to do genetically modified mosquitoes. When I asked about it, nobody ever told me that this was part of a study. So that there is secrecy and there is non-disclosure here. I was a confirmed dengue case, and I don't want to have it again. And, but I would like to see certain questions addressed. For instance, over 3 million mosquitoes were released in the Caymans, and 3 to 4% are judged to be fertile. You know, the males can have offspring, and these offspring can mate and be in the air. That means that 3 million mosquitoes, that means that 90,000 genetically modified mosquitoes could have reproduced in the wild there. So this is an invasive species. Non-indigenous. What are the impacts of this? And we don't know the health risks because it's so new. Why not study really what's happening in the Cayman Islands? 0.5% of the females released are in fact females. You know, this is, you know, the company sexes them by hand. I wonder how I could sex 3 million mosquitoes safely, but that means approximately, if we're taking the least amount, 15,000 females were released. Since females bite humans, with the impact on human health for citizens, and what are the health facts for the Cayman Islands? What happens in the food chain if genetically modified progeny outcompete other mosquitoes? Will parents feel comfortable with the possibility of their children being bitten by mosquitoes? Will tourism be affected? I don't think you've researched this enough, and I think you've done it in secrecy. And as a taxpayer, who will be financially responsible if we are sued for releasing these mosquitoes and it backfires, we're in trouble. I think it needs to be shelved, put aside, and studied to make sure it's safe. And there are places where these mosquitoes have been released, and they're not going to go away, some of these mosquitoes.
Thank you very much. Yes, could you cover the questions regarding the numbers and the releases?
So thank you for those questions. And time is limited, so my answers will inevitably be brief. I'd be happy to discuss this. Sorry. Okay, so time is brief, so my response will inevitably be fairly brief. But of course, these are questions considered by regulators and could be discussed at more length later. But a degree of incorrect information has been put out about some of these things. So let me try to cover a few points. Let me see. The female release, we were not releasing 0.5% of females. We were releasing about 1 female per 1,500 males. So we do inevitably release some females. So then you have to say, what is the consequence? The numbers are far lower than you were indicating, but not zero. So then what happens if you are bitten by one of these mosquitoes? We rear millions of these in our laboratory in Oxford all the time, and of course we are rearing them in the presence of tetracycline. So we're breeding them, so we have to have the females, and so we have both analyzed them in theory in terms of what proteins are being produced and how they're being expressed, and they are essentially the same as a normal mosquito. The modified proteins that we produce are not detectable in the salivary glands, let alone the saliva, so they're not injected into people. And furthermore, from a more sort of human or anecdotal thing, I have been bitten repeatedly by these mosquitoes, and so the people in the laboratory, and they are the same as a normal mosquito in terms of being bitten by them. They are actually a little short-lived in the wild because of the modification, and that makes them substantially less able to transmit dengue or other viruses that might be around than a regular mosquito might be. But otherwise, in terms of what happens if an ant or a scavenger or a bird or something eats one, or if one, to the very small extent that we release females, they are essentially the same as a normal mosquito. What else can I say? The populated unpopulated area thing. I think that's a confusion between different trials. So in Malaysia, the release experiment was in an unpopulated area with a lot of information with the community and signs around it and that kind of thing, but an unpopulated area. In the Cayman Islands, it was in an inhabited area, and also in Brazil, where we're releasing at the moment, in an inhabited area. This mosquito is very human-associated, so populations of this mosquito, Aedes aegypti, are always around humans. They don't fly very far away from humans. They bite humans, and they breed around humans. So if you want to try to control an Aedes aegypti population, that population will inevitably be around humans. And so the exception, if you like, was the Malaysia experiment, which was in an uninhabited area. And there we were looking at how far these mosquitoes fly compared with an unmodified laboratory strain. So a side-by-side comparison of two strains in the absence of a wild population of mosquitoes. But that was a somewhat exceptional experiment. I'm sorry, I have some notes of your questions, but I may omit some. Oh, the survival aspect. So in the laboratory, under ideal conditions, ideal for survival conditions, so when we're very nice to them, indeed 3 or 4% of the offspring that inherit one copy of this gene survive through to adult. Those are weak and short-lived. In the field, where we have done this sort of experiment with some other insects that had similar traits, actually a higher survival rate, we found no survival in field conditions. Life is just tougher in the field than it is in these ideal laboratory circumstances, and so we would expect a much lower proportion of these insects to survive to adulthood in the field than that 3 to 4%. Where we have looked for that in the Cayman Islands particularly trial, we found no survivors in the field, but the numbers that we can recapture are not so large that you could definitively say it was zero, but in fact we found none. And then, indeed, when we stopped releasing, so this question of can it establish, or it's sometimes phrased, is it an invasive?
Luke, if you could wrap up in just a moment to give more time.
In terms of, is it an invasive species? At the end of each release period in the Cayman Islands, for example, of course, we monitored what happened to the transgene in the wild population, and indeed it disappeared rapidly after release. So the males that we released die, and that's within a few days, and they were undetectable in a week, or I don't have the exact numbers, but within a matter of days. And then the females that have mated them correspondingly die, and so the transgene was undetectable in the region in a matter of a few weeks after we stopped releasing. So no, even though we released several million mosquitoes, the population did not establish and disappeared rapidly after we stopped releasing.
Okay. All right, thank you. Okay, we're on to Judy. Let's see. Judy Martinez. Right here.
My first question was answered. Do they bite? The answer is no. My second question is more political, as usual. Could we bring this before the City Commission and put it up for a referendum for a vote in November to see if the city in general wants this?
I could certainly talk to them about it, but I don't think, because we cover the whole county, the city is just one of the areas in that.
I take
I'd be
it that
happy to
most
talk
of the
to them.
people do not. That's my general impression.
In terms of thoughts about the what we've done so far, Mickey, can you give us any information in terms of responses in the field from your inspectors and when they've been talking about this?
The majority of the people that the inspectors have talked to are indifferent or just want more information. We've had a couple very negative responses to it, but majority of it just wanting more answers. So we don't have that real negative. Right,
right. And the goal of this is, and very honestly, is the people that make these decisions. I want to be people who have the scientific background to evaluate risk, and that's really what this is all about: is the risk of any future mosquito-borne disease worse than the risk of a new technology like this. And as the director of the mosquito control, my job, our job, is for the whole district is to protect people. We would not do anything that we felt was harmful. That's the point. Everything we're trying to do is to find the most healthy environmental way to suppress potential issues. I saw that when I was in, well, I don't want to give too much of my own time. I want to give more time to everybody else. Okay, thank you very much. Okay, now we have Jody Smith Williams, and then after that will be Oliver Kofoid.
Hi, good evening. I would like to know why the request by some organizations with some experts with opposing opinions, alternate opinions, were not allowed or invited, I should say invited or allowed, to be here tonight. I think, we've had a great dog and pony show from the people who have everything to gain from this project going forward. And I've done my best to try to educate myself. I'm not a scientist. Probably most of the people in this room are not scientists. There's many questions, many that have been stated already. But I think there's questions and debate that should happen from people that are truly qualified. And you mentioned the regulatory agencies, okay. But where are they, this evening? This is supposed to be a public forum where we can get all this information out.
Yeah, as I mentioned in the beginning, it's a gathering information about what the issues are, because we're still pretty early in the process. Yeah. So for a forum at some later point, I want to know who should I bring? Genetic people? Should I bring population ecology people? What that's part of what we're here. I want to get a cross section of what people are interested in.
Well, I'm happy to hear that, and I hope that you'll follow through on that. It seems that, as many people have said, this really has been shrouded in secrecy. It's been in the plans behind the scenes for a very long time. and reports last year that were in the news media, not too much in our local media, but in other media, saying that releases may have happened as early as January. I don't think that's happened. I believe you that it hasn't happened yet.
There's been no.
No, I understand. I believe that, but I'm just saying that that was the plan, and yet there was no urgency. We should, if that was the case, if that was ever going to be the case, this meeting should have happened many, many months ago. So now I feel like there's a lot of backtracking and trying to put on a good face, and now the delay, the process has been delayed. Thankfully, I appreciate that, but it undermines I think, the public's confidence in the whole process. The fact of the secrecy that's gone on so far. I would also like to know, back to the very first question tonight, of who ultimately will be making this decision. Yes, the regulatory agencies, if we ever figure out who that might be. Are we committed to having some kind of regulatory agency sign off on this, first of all? And then within mosquito control, is it a board decision? Is it an executive director decision? I feel that you have been at odds. You've been more cautious than the board has been. At the last meeting, and in the headline in the newspaper, they said we'll go forward with or without permits. So how do you answer that, please?
Okay. So to answer your okay, Tommy. To answer your last question, the decision about should it shouldn't be done, I want to get regulatory oversight for this. I'm not a geneticist. I do have genetics folks on our staff. Yes.
Okay.
And in terms of a decision, it's the decision to do it or not here in this district is ultimately my decision. It's an operational decision. So that answers that question. And then to do one other one, did I miss?
Can I also?
Okay, go ahead.
If I can also make a couple of comments on that. We, Oxitec, really want regulatory oversight. We spent 3 or 4 years talking to federal regulatory agencies to try to find what the regulatory pathway is going to be. So not specifically about Key West, long before there was any discussion about Key West, but just in general about the regulation of genetically modified mosquitoes in the United States. Okay. So, and we absolutely want regulatory oversight and everything that goes with that. As we have, indeed, and as we have had in all the other countries in which we have operated, we think that yes, absolutely, the independent evaluation of technology is essential for public confidence in it. Why would you believe what we say without external reference, as we are interested parties? In terms of the timing, and I'll comment on this because I think this sort of January comment may have come from me originally. There's a difficult trade-off between telling people what we have in mind at the earliest possible stage and having something reasonably concrete with which to go to them. So if we have some sort of ambition to do something on a particular timescale and we say so, that may seem like a definitive plan, and it is not. But on the other hand, if we have only sort of airy-fairy general ambitions to do something in this sort of general direction, then it's quite hard to come forward and talk about it. So there is a trade-off between, having something reasonably concrete to bring to you and the timing thereof. And so I was in that comment where I once upon a time said January, about a year or more ago. That was in response to a question: what is the earliest time you could, from a technical point of view, possibly go forward? Something along those sort of lines, and then it gets put out as a definitive plan, which it was not.
Right. Right. And if you look up here real quickly, just in terms of that, we saw this slide before about the population and the numbers to be released. I'll jump through that very quickly. Ideally, if you try to release 10 times the number that you that are presently out there. So early in the season, say in March, if you want that you'd be very few we need to release. For financial reasons and manpower, that makes most sense. If you try to do the same release for in say late July, it'd be 10 times that number. So you can see that it's based on you want to overwhelm the males. In the fall, you can do small-scale releases, but the ideal time, if you look at this, if to do it, would be about a March time frame, ideally. And so if that So in terms of January, it's like when would you want it to get started? If you would get started in January, get things figured out and everything in place to do a release a little bit after that. So that's why some of the timing things came up. Okay, now on to Oliver. Is it Kofoid? I'm sorry if I've. Very good. Thank you.
Oliver Kofoid, Northside Drive, Mosquito District 1. Some of my questions, because I am a citizen and I also am somewhat of a scientist. I'll start out with Oxitec. How many contracts do you have federally, abroad, and with those contracts, are they permitted? And so far from what I've seen, it's been with 3 places that you have done. As a scientist, usually we like a lot of data over time, and 3 data points with the releases there is kind of skeptical. And I would like to see a lot more data on that. Before I would even consider this, I would also like to see a tetracycline study done in Key West before anything happens. Because I also work in the wastewater field, there's a lot of pharmaceuticals that come, and I realize you're saying 1,000 yards for your treatment area for each release. Well, with such a small treatment area, I believe the Mosquito Control Board, with what they have, and the mosquito professionals that are out there can very well treat that in that area. I mean, look, we didn't have any outbreaks last year. So to reach out, I realize that you guys at Mosquito Control would like to have this in your back pocket. Now, is a federal agency going to permit it? Is a state agency going to permit it? Or can the Mosquito Control actually permit with Oxitec if no one comes back? I'd like to know that also from Mr. Doyle.
Okay.
That's about all my questions for now.
Okay. In terms of regulatory, we're not a regulatory agency, so we would not issue our own permit. We work under the Department of DACS, Department of Agriculture and Consumer Services in Florida, and then federal agencies as applies. In terms of tetracycline, I actually have looked into testing. I've looked at University of Florida and Mote Marine Laboratory to get some information. It looks like for 100 samples it would cost $18,000 to do that, and I'm hoping that something like that would be part of the of a review process. In terms of from our staff looking at the data that we have, the real question is, do Aegypti grow in places that have tetracycline, or could? The real answer that we've seen from our data is here in the Keys, which is different than work in other countries, is really no. I think I mentioned before we had 2 out of almost 4,800 samples that we found with it. Can you find Aegypti once in a while in, say, a sewage treatment plant? Yes, occasionally you can, but it's I think believe 0.04% of the ones we find.
I also have something for everyone up there. I'm wondering if you have heard of this Operation Bellwether, and if you have you checked out the congressional reports? And I would say to anyone here to go on Google and check that out, and go to the congressional reports to check that out.
Okay, thank you very much. Next we have Connie Tarpley, and after that Dan Mastracchio, I believe.
I'm Connie Tarpley.
Connie.
Actually, I think a lot of my questions have been answered, but I would like to defer to my friend Ms. Jolly, who said she had lots of questions. So if that's permissible.
You want to give her your 3 minutes?
Yes, please.
Sounds good to me.
I liked what the other guy said. I trust the clock is starting now. The first thing I would like to ask is, Director Doyle, when these further public hearings will be held, will the folks who carry direct opposition to these methods, will they specifically be invited to participate? You can think on that. I have a lot more questions, and I
Go
want to
right
get them
ahead.
into the record.
Yeah.
They were, they were.
But I noticed you stalled.
They were told about this meeting. You're talking about Friends of Earth and Gene Watch and all these groups. They were told about this meeting, and we've had an ongoing dialogue with them almost on a daily basis.
Thank you so much. I'd like to get the rest of my questions into the record. For the gentleman from Oxitec, is this your document?
I can't see it
Key
from
West,
here.
Oxitec Q&A? Key West,
Q&A.
Oxitec Q&A. This is your document. On the last page, you say that you have a community engagement plan in place. This is a question of integrity, and please bear with me. You say that you have conducted so far have consisted of public information events. Where have those taken place? And has anyone in this room been to one?
Okay. Yeah, it's like if I think that, are you finished or?
For the record, nope, I'm good.
Okay.
I'll keep
All
going.
right,
If they
I'll keep
can't
notes on
answer
that
the
one.
question, I'll keep going.
I'm willing to answer.
We were looking to see who's going to answer the question. Colleen?
I do all the public outreach through for mosquito control, and I've talked to the county commission. I've talked to Key West City Commission. There's been articles in the newspaper. We're on the radio every week.
Public information events
Are you talking about
indicate
a
being
specific
out
town
like
hall
this.
meeting? Yeah,
Like
this is our
a
first town
public-
hall meeting. This is just the beginning, though.
So for integrity's purposes, Oxitec states that to date, community engagement activities so far have consisted of public information events. I just wish to say, gentlemen, I want to trust in your highest intent as scientific people, not dollar-driven. But please understand that when statements like this are in your own document, it gives us pause. I'm not done with the questions, though. I would like the regulators who will review this DVD to be sure to address the inconsistencies in the scientific responses. For instance, genetically modified females essentially the same as the wild. That's a wide range, essentially the same. And why, can someone say, why has a trial not been done in a very controlled faux human environment rather than making the populace, the citizenry, the human beings, the actual guinea pigs? And if we were, let's just say everything goes and despite our resistance, this does take place, what kind of commitment are the citizens of this county making to be in relationship with Oxitec and these genetically modified oxymosquitoes?
Okay, can you give a very short answer on this?
Okay, in terms of just to, it's very difficult to pick on all those. The genetically modified females, essentially the same as wild type. They have a specific piece of DNA inserted, which is completely characterized and sequenced. It has 2 genes on it, and we know the properties of those 2 genes. Apart from that, the backbone is the same. That has the particular traits described, I think, in the Q&A, but certainly in the scientific literature. So they are, other than the modification, essentially the same as wild type ones in other respects. So a legitimate statement, I think.
Do you mean in terms of lifespan?
Yeah, absolutely.
Food they eat,
life
number of people they
history
bite?
characteristics, composition,
Just wanna
behavior, interactions.
If I can interject in that, because ones that are reared in the lab, if you take any wild species, do not change the genetics, put it in the lab for several generations, and then put it back out, it'll live a little bit longer. They're not bred as tough.
Sure.
And that happens with any. I reared when I worked at CDC. I reared a lot of mosquitoes, and that happens with all species, regardless of any genetic.
But none of us are scientists, so please be as clear and accurate as you can. Thank you.
Yeah, so, okay, I think that's.
Yes.
In terms of testing, we have tested in indoor environments of one sort or another. There is a limit to the realism of an indoor environment, so there's a limit to what you can learn from that. But to the extent that we can learn that, we have done that. So we have done it in small cages, which are probably not very realistic at all, but you can get big numbers and solid data or something that is not that realistic. And then the closer you get to the field, you're doing smaller numbers or something more realistic. And so, for example, we did some large cage studies. Our Malaysian collaborators built a simulated apartment to look a bit like a apartment in Kuala Lumpur, and did mating competitive studies in there, and there was a human in that behind screens, because mating behavior is affected by human odors, so that is rather the sort of faux human environment that we're looking at. And we looked there to see whether our mosquitoes had a reasonable chance of competing for mates with wild ones, and they did. And so in a set of sort of incremental tests, that was one step, and before that were small-scale laboratory experiments, and after that we were going on into the field. So the sort of thing that you're talking about, which is broadly a sort of incremental, step-by-step testing process that goes from small-scale artificial circumstances through to the field, that is exactly the procedure and philosophy that we have used in testing this strain.
Okay. We do have 5 more questions to go, and I'd like to move on if that's okay. This is Dan Mastracchio. If you can raise your hand. Dan. There you go.
I only have 3 minutes, so my first question to Oxitec, I'd just like a yes or no answer to, please. Did you use the herpes simplex virus in the creation of this mosquito? Based on the Cayman Island risk assessment, that's what it says. Is that true, yes or no?
We have a segment of DNA from, which is called SV40, so it's not a gene, but it's a regulatory element that is originally derived from a herpes simplex virus. It's a very well characterized element, widely used in genetics and genetic engineering.
I'd like to, based on the idea that Aedes aegypti would be contained because they only travel 100 yards, anybody can drive their car anywhere near the 100 yards and a mosquito could get in it and drive to Miami-Dade, so on and so forth. I think that's a rather ridiculous statement to think that it's not going to get into a car. Probably everyone in this room has had a mosquito in their car and it can go anywhere. My next question would be to the Mosquito Control Board. You mentioned that tetracycline is not found, predominantly in the Keys, but it's found in drinking water in most places. It's found in, it's one of the most commonly used veterinary antibiotics. It's found in the feces of animals. It's used as a livestock promoter, growth promoter. It's found in sewage sludge, as was mentioned before. It's very commonly used, and the idea that it's not going to be, anyone could take their dog to the vet and get some tetracycline, and the main way that it's used is to be put into water, so they can leave their dog dish outside with tetracycline in it, and a mosquito can go in there and lay their larvae, and now you have genetically modified mosquitoes everywhere. My next question would be that why hasn't the Mosquito Control Board attempted to use ovicidal and larvicidal traps with attractants similar to those used in the Philippines, which is basically one of the things that you mentioned is that you have a difficult time controlling the containers outside that people are using. Well, by using attractants and larvicidal traps, what you do is you actually attract the mosquitoes to those areas where you're not having them land as often in the containers that you have outside. And not only that, but studies suggest that when Aedes aegypti lay their eggs in a specific spot, that they continue to come back, and it actually, the eggs itself in the area that they laid it is actually acts as an attractant even further for them to continue to lay their eggs in that area. By using ovicidal larvicidal traps that are extremely cheap in the Philippines, these things cost approximately 20 cents in the U.S. per trap, and they cost 2 cents for the pellets, which would last a week for 4 traps, which is recommended. That equates to about $2 for the entire year per household for ovicidal larvicidal traps. That it goes beyond what the Mosquito Control Board is able to do with helicopters using larvicides from aerial sprays and so forth. This acts on an individual household basis where
You just finish that statement, then we'll start answering.
It acts as a household basis where you're not going to be able to spray larvicides inside of someone's house, but you can with an ovicidal larvicidal trap, where Aedes aegypti is most likely to attack someone anyway, is going to be in their home or in an area around their home, which is specifically where they'll be protected by the ovicidal larvicidal traps.
Thank you very much. We'll get to that. Okay, so let me just answer 2 questions first. The one is tetracycline in the environment. 2 things I can say. Aedes aegypti prefers clean water. They don't like sewage water. You don't find them in sewage water, or very rarely. You can never say no. They never in science hardly. But everything that we've looked at, Mickey, just a yes or no. Do you know if any of your inspectors have ever found aegypti in, say, dog dishes?
Yes, when they're not maintained, they are found in dog dishes. Most people do dump out their dog dishes daily, give their dogs clean water. But yes, when you leave your plastic containers or dog dishes out for a week at a time and do not change its water, yes, they find it all the time.
So that would be a good place to take a water sample, in my opinion, because that's a likely place in an urban area. The second question regarding the ovicidal traps, I appreciate you looking at alternative methods, because that's excellent. Because just doing the same old for mosquito control isn't going to work in the future. In terms of ovicidal traps, Andrea Leo is going to just briefly answer about what's been tried here with that.
Well, many of you guys know about the lethal ovitraps that we put out last summer. Actually, it was 2010, and it was basically a fiasco. We found a lot of those ovicide, or the ovicups, actually breeding mosquitoes. So because that technology was so new, when we were doing this trial that summer, it did not impact the population of mosquitoes whatsoever. But again, it is important. We're constantly looking at new tools and trying new things to see what will work best in our environment.
I think we put about 7,000, isn't that right?
It was, yeah, it was about 5,000.
5,000
lethal ovicups. No. Which is
sure.
Right.
Tell me about it. Very honestly, right. I mean, I really like those alternative controls. In fact, when I first got here, I thought maybe we could try. It's a sugar trap, essentially. You put a small toxin in a sugar bait, and mosquitoes go to it. And then I learned very quickly that Aedes aegypti doesn't take sugar very often, because it gets most of its sugar from its blood, unlike other species. So unfortunately, it wouldn't work with aegypti. I was disappointed with that. But go ahead, Luke, very, very quickly.
We would see our technology being used as part of an integrated program with the best available methods, and lethal ovitraps would be a good combination. Unfortunately, the ones that are around at the moment don't work very well. I was in the Philippines not very long ago. They are very interested in our technology because the methods they have at the moment do not work. But if if other such technologies were developed, that would be excellent, and things that kill larvae would be ideal, eggs, larvae. Regarding tetracycline, it's come up several times, so I think I probably better give some numbers. So our mosquitoes need quite a bit of tetracycline to survive, so you see no excess survival at 10,000 nanograms per litre. I'm sorry I'd have to do this in numbers, but the numbers are really important. We have surveyed the literature. A lot of people are interested in contamination in the environment with antibiotics and indeed other things, and so there are surveys from around the world and around the United States of tetracycline concentrations in sewer water, in input to wastewater treatment plants, to output from wastewater treatment plants, and water bodies of all sorts. And we have never seen a report. We have surveyed the literature as extensively as we can, and we have not seen a report of a concentration of that level, 10,000 nanograms per litre, in any kind of water body, and that concentration is a level that would give no excess survival. Furthermore, not only have we not seen it, but people who have raised this kind of objection, like Friends of the Earth and Food and Water Watch, I have been out to talk to them, and I have asked them, because they are also interested in contamination in the environment, to provide us with any data that would indicate that level of tetracycline concentration in any water source, and they have not been able to do so. Furthermore, if you were to look for that, if you were looking for tetracycline, you would look in highly polluted water that is not where Aedes aegypti are breeding. So first of all, there's a spectrum of water from sort of clean to dirty. The Aedes aegypti are absolutely breeding in the clean end, but even the dirtiest end of the spectrum, as far as we can tell from the literature, from other people's surveys for this kind of thing, not ours, does not have a tetracycline concentration that would be sufficient to give survival of our mosquitoes. And that's not too surprising, because tetracycline is relatively unstable. It's sensitive to light, so it's degraded by light, it's degraded by temperature, and it has a relatively short half-life in the environment.
Okay. Let's move on to John. Unfortunately, we have to be out of this room at 7:30, so I'd like to finish up at 7:15 to give us time. So let's move to John then. And after that, Rick Worth, and we might be out of time at that point.
I would like to know what peer reviews that you have that's not funded by your company. Also, are you funded by the Bill and Melinda Gates Foundation? And I read somewhere that your company is losing $2.7 million a year since it's been founded. You obviously have many investors, one of them being a Boston banker that you're set to pay back a debt to at the beginning of 2013. Just want to know, is that true? And that's it, just about those questions.
Okay, so in terms of peer review, we publish in the scientific literature, and therefore in the peer-reviewed scientific literature, pretty much everything that we do of any note. And so I would say to an extraordinary extent for a small company, actually, probably because of our and indeed my own personal academic background, we do publish what we do, and we do that in the peer-reviewed scientific literature. My surname is Alfie, and it's relatively unusual, so it's quite easy to find those papers by database search. And so indeed our collaborators also publish. Those are perhaps a little harder to find because there's no. Well, when we provide, people who want to work on this come to us for the material on a collaborative basis. So sort of inevitably, and as is normal in research science, those end up as some sort of collaboration. They're still done independently by independent scientists working in another institution or laboratory. And so, so the Cayman Islands trial, which we've published, the lead author on that paper is from the Cayman Islands government Mosquito Research and Control Unit, and not one of us. Although, of course, we were also involved, and so we are also authors on that paper. It's very, very normal scientific publication process. The unusual part of it is that we publish so much of what we do for a small company. In terms of the Bill and Melinda Gates Foundation, our funding. We're part of a consortium led by Tony James, member of the National Academy based at University of California, Irvine, going back to independent review and collaborators. And that consortium is funded by the Grand Challenges in Global Health Initiative, which is a big funding initiative, which is itself funded primarily by the Bill and Melinda Gates Foundation, but also the Wellcome Trust, Canadian authorities, and so on. And again, through a sort of peer review process for the grant stream in the usual sort of way. Do you want to talk about the finances? I mean, our financial situation is very typical for a small company developing a new technology, but Hayden can address that.
Yeah, sure. We're financed entirely by private investors, and they put their money in principally through investment as equity. And yes, we do have a US investor, and they've invested in equity mainly, and they've also and they also put in some money the year before last as a debt, as a convertible debt, because that's what they chose to do, and that's how we're financed. But we're not. Just to answer another question that actually came from a gentleman at the back. The way we work, just to be very, very clear, is the way we're working with Florida Keys is what we basically say is yes, if you'd like to go ahead, then we'll supply our time free of charge. We'll supply the mosquito eggs free of charge. We'll supply everything, and you can evaluate it and see if you like it or not. And if you do, that's great. And if you don't, that's great. There's no contract. No one's paying us at this point. I mean, this is an approach which people need to see if they like or don't like. And it's the same in several different countries.
Okay, if I could add one thing and then go on to the next person. We in terms of ongoing relationship, the relation with Oxitec would be the same as it would be with anyone else who supplies us mosquito traps or mosquito control pesticides or anything else. That in order to do mosquito control, we'd be buying things from some company somewhere to do the job that we need to do. And so, and that would be the same. We'd be buying, if this happened, we'd buy eggs versus bags of product or machinery. So that's, now we need to go to Rick Worth, and this will be the last question, unfortunately. I'm sorry, the which question?
He answered.
Yeah. I believe that was the one in Boston. In Boston, I'm sorry.
Yeah.
What I have to say, Mr. Doyle, is very clear, and I'm very disappointed at the way this forum ran out today. First off, the good people of Key West and Monroe County, they gave you a voice, not to take our voice away. If we in the majority would like to say no to this whole thing, whether you've already signed some agreement with them or have taken a down payment, a majority of us, if you even took a hands by this group that you say shouldn't be taking and making decisions because we don't have the scientific knowledge, I don't care. It's an absolute no for me, and you've done nothing to prove your points, and you've done nothing but shield yourself and to tell us that you're going to shove this technology down our throats. And I, for one, don't care about your scientific crap. I don't care about money you spend. You're not going to cram something down my throat that I don't want. I'm no guinea pig.
All right.
Yeah, I got more time.
It looks like we've got.
We're at 7:15, I would say.
What's
We
that?
have
I have three more people. Kath. Okay, let's do it. Very good. Catherine DePoe, thank you.
I actually don't think I need this, but anyway. I think I have a misconception. This lady over here said that she had a question whether or not the females bite, and the answer was no. I understood they do.
Yeah, the females bite, the males do not.
Bite, so they're
Yes.
just the same. Okay. Has there been a problem getting a regulatory agency to agree to oversee this? And in the absence of getting a regulatory agency to oversee it, are we just going to march ahead? That's one question. And then during the same period of time that the dengue fever knocked off, do we test for actual mosquito population? So has there been also a drop in the levels of mosquito populations that we could attribute to better education that is going out to the community? Because those two things kind of happened around the same time. So absent a regulatory body agreeing to check and assure the science, perhaps we're better served by increasing the education, which is a long-term benefit, because it seems to be working, and it engages the community on a whole scale, as opposed to everyone kicking back waiting for the mosquito board to take care of this problem for us. So those were my basic questions. Has the population dropped? Are we going to march ahead? Are we having a problem getting a regulatory agency to address this issue?
Yes, given the time, unfortunately, we would be out in 14 minutes. We have because we rented this room for that line. And then, so what? Thank you, and I will. We've got that on record, and I will answer those questions on the website. I'm sorry to do that, to put you off right now, but I want to hear everybody else. Okay, next is let's see, Jacobs. I can't get the first name.
Encouraged by the county and Oxitec statements that they are dedicated to be less dependent on pesticides, since for the last year we've been smogged with a cocktail of chemicals with no input from our community, and just scientific names. I have a strong autoimmune system, I believe, but I have noticed that after these smoggings I've experienced sore throat and oral nasal discomforts. I do believe that they're related. Further to that, the concept that we're playing God with genetics. We know that our environment is an intricate web of life, and that genetic adaptations will normally take thousands of years, if not tens or even hundreds of evolutionary years. And we're dropping these new genes into the environment with no real knock-on effect study. What will be the long-term effect through the food chain? Animals that eat those bugs. What are the considerations for mutations down the road? I think there's been very, very little real study and a lot of conjecture. A lot of, well, we believe. Believe is very far from the facts. I believe we need to do a lot more study before we go ahead with a movement like this, and particularly given the note that the numbers are on the decline. Don't we have time for further education, seeking healthier, more symbiotic relationships that are going to help us with this issue? What about the introduction of, rather than breeding more males of these robo-Franken mosquitoes, why not introduce dragonflies, natural predators, things of this nature that aren't going to enter our food chain and damage our fish, the food that we eat? I'm tired of eating chemicals. I'm tired of being showered with chemicals. I think it's time for our representatives to stand up for us and stand up for all life, and stop with this nonsense. We're seeing knock-on effects in genetically modified corns. There's beliefs now in relation to cancer cases. Many other things that we're seeing in our environment. Massive populations of bees dying, possibly related to genetically modified agriculture. When is this going to stop? When is life sacred? Thank you.
The final individual will be Dr. Bill Irwin. He had to leave.
Apologies, he's on our science team for the
Okay.
Florida Environmental Coalition. He
Okay.
asked me to ask this question. His question is, as a scientist,
We just want to get it on. You can go ahead and do that, but we need just we'll quickly have to do it in 3 minutes. Thank you.
absolutely, it's very quick.
Tell me when you're ready.
Go ahead.
Okay. On behalf of Bill Irwin, PhD, he's a scientist on our science team of the Florida Keys Environmental Coalition. On his behalf, he wanted me to ask this question: scientific questions about the genetics, the specificities regarding the genetic alterations can be directed to whom and where?
Okay. The answer to I'm sure the folks at Oxitec, Luke.
Genetic question.
Yeah, absolutely, of course.
Yes.
Can be directed where in the future, not just tonight, but what contact resources can you offer?
Yeah, absolutely. So I would certainly encourage anybody with any kind of question actually to send them to Oxitec. But of course, you may also wish to send them via these guys to the regulatory authorities, who would also then consider the questions. But I would be delighted to receive questions, especially about genetics, but on any of these topics, direct.
Would you be willing to give us a website or a name, something?
Sure. So our website is www.oxitec.com. Oxitec is OXITEC.
I have that already.
Okay. And there's a contact us on that. My name is Luke Alfie. Luke is Luke. Alfie is ALPHEY.
Right.
And is that Luke.Alfie?
Yes. Yes.
Yes. Okay. And I'm sure there's geneticists and others at universities. We could probably get a list of people.
Yeah, absolutely.
Who would be interested. Who from all ends of the spectrum in terms of their views on this topic.
Experiment in Mexico failed, and that the females are breeding. So.
Okay. The answer to that is no, just to make that's
Yes,
a short
we're
one.
going to
Thank
find a
you.
regulatory agency.
Okay.
We spent a lot of time doing that.
All right, so I'd like to make a couple quick closing statements, and then if you wish to speak afterwards, I'm certainly here for quite a while to do that. So thank you very much. But we need to do it outside the room. Before I forget, we have the anonymous survey that was done by North Carolina State. Michael Cobb is in the back, and Michael, how would you like to do this? Okay. Okay, again, thank you everyone for coming and for providing your information.
You've wrapped it up nicely.